Nucleoprotein, 3 domains, one at the front, the N-terminal or amino terminal domain. You'll see it as NTD on here. A middle domain, which tends to be full of serine and arginine and sometimes asparagine, which is N. A C-terminal domain, which is again folded up in like a little ball. The C-terminal domain is going to help the nucleoprotein dimerize. And the N3 domain, the mysterious N3 domain, that's super important for packaging and budding interactions. This is going to be the only part of N that directly interacts with the M protein. M as in M, upside down W protein. I know M and N and they all sound the same when you say them, when I say them. All right, so what does N protein do? We think N protein is going to wrap RNA. So N protein is a dimer, and we think the RNA probably goes around the outside of that in some way. Nobody has yet solved the structure, so we can't actually show you a picture of it, so we don't know. This up here, that's a picture of a spike protein. The little blue things are the glycans. They look great, don't they? What we do know, though, is inside the virion, N protein ought to form a helix. Usually RNA viruses, you get a long helix of N proteins with the RNA either inside that or outside that, but not this time. In this case, you get a bunch of small helices, and we think the RNA is probably going somewhere around the outside of it, but each of these helices is going to contain several nucleoproteins. Here you can see these little blobs, so each one of these is probably an N dimer, and there you can see them pointing in, they sort of touch at the center. That's probably a little part at the N-terminal domain of the nucleoprotein. So C-terminal domains on the outside, N-terminal domain in the center appears to be the conformation. Somebody may solve a structure in a year's time that completely reverses that understanding, but that's how we understand it now. And that's all you can teach, is now. Okay, what does N do, and how does it do it? N is going to be involved in packaging. And remember, packaging is interacting with the genome RNA, not just with any willy-nilly with any RNA, but with genome, special genome. And the genome RNA is going to have this thing called a packaging signal. We're going to abbreviate that as PS for packaging signal. This is the packaging signal of mouse hepatitis virus, or MHV. It's another beta coronavirus. It's pretty close to SARS-CoV-2, although it's in a different subgenus, but they're in the same genus, beta coronavirus, so not too far. We're showing MHV because we don't know how this works in SARS-CoV-2. It's another embarrassing thing we don't understand. Man, why is virology, why are we so bad? I don't know. Terrible. But you can see on this packaging signal that there are repeating units with the little 2 A's or an AG hanging out and the CG-GU-AU pairs that are up here. That little unit seems to be just about equally spaced here. And if you go in and mutate individual residues of the N, if you change any of these lysines that make sort of a ring, so the blue is one copy of N, the green is the other copy. This is the actual dimerization. This is the point of dimerization in the dimerization domain. This is all just the C-terminal domain that you can see here. But you can see between 2 N's, you get sort of a ring of lysines. Those rings are presumably what's going to be interacting with these. Are they sticking through the middle of that little bulge? Are they grabbing onto the edge of the little nub that sticks out? I don't know. They're probably interacting with the backbone of the RNA because lysine is positive and the backbone is going to be negative. Beyond that, beyond some kind of salt bridge, I couldn't tell you. But N is not done with its little tricks that are involved in packaging and assembly, because there are also packaging interactions that are going to occur here. So there's one amino acid in the M protein. It's a lysine that's conserved. And there are several acidic amino acids in the C-terminal domain. So it's the N3 domain, is what we call it. It's right at the end of the N. And all of those are involved in specifically packaging genomes. So they're also somehow involved in touching this great big structure. How? Why? I don't know. Definitely don't know. Hasn't been shown yet. Someday that'll make for a very interesting paper. For now, we just don't know. All right. N is also involved in interactions with the M protein. It interacts with just down here at the tail end of M, and it seems to be these two aspartic acids, so they're going to be negatively charged. The arginine is going to be positively charged. Those interact in the M proteins that we understand. We don't understand a lot about M, and we don't understand very many M's, and there's a lot of different stuff there. So this is the way it works for SARS-CoV-2 and for some other beta coronaviruses. It may work a little differently for other coronaviruses, like maybe the acidic amino acid is here and the basic ones are here. It would still make the same salt bridge. The virus probably wouldn't care, but there's no way to tell from alignments. All right, and that has been the story of N. Hope you liked it.